Good afternoon, everyone, and welcome to another presentation in the TRANSCEND Seminar Series. We're especially fortunate to have Dr. Jefferson Vaughan with us today, presenting oral medications prescribed for flea and tick control in dogs have potential to augment malaria control. Dr. Jefferson Vaughan earned his PhD in entomology from Virginia Tech and specializes in the transmission of human and animal diseases by arthropod vectors, such as mosquitoes and ticks. His research spans a wide range of vector pathogen systems, including malaria, arboviruses, avian filariasis, neorickettsia, fleas, and ticks. Dr. Vaughan's current work focuses on avian malaria and the development of innovative vector control strategies, including the use of systemic parasiticides in livestock and rodents to reduce disease transmission. Please join me in welcoming Dr. Jefferson Vaughan.
Yay. Thank you, everybody. Thanks so much for the great invitation. It's great to be here. I'm excited to be here, and thanks for everybody for turning out today. So this is, of course, part of the TRANSCEND program, and for those of you that may not know, the goal of this program, of course, is to bring together basic researchers with clinicians, okay, so that we have the opportunity to help develop innovative means of combating diseases in North Dakota and maybe even beyond. So my purpose here today is to promote the repurposing of the latest generation of flea and tick medicines towards human use to help control mosquito-borne diseases. Okay? And for me, the crucial step in this process is trying to do some phase 2 human clinical trials. So that's my purpose in coming to the TRANSCEND organization. Okay, but before I get there, I want, research doesn't happen in a vacuum. You need funding, you need people to help you do the research. So I have a few shout outs here. Some of my funding sources, some of my early work with the ivermectin was supported with an NIH R21 grant, a little exploratory grant. But most of the work I'm going to tell you about today was funded by a pilot project grant through the through the Host Pathogen Interaction COBRI group, okay? And that effort is led by Doctors Dave Bradley and Catherine Brissett. I also have some graduate students now that contributed to it. Jumi Adewale was a PhD student from Nigeria, so she has a vested interest in what we're doing. And a lot of what I'm going to be talking about, I've had tremendous help from my two master's students. They're here in the audience today. Tyler Sternhaus from Wisconsin, I believe, and Zane Seidel from New Mexico. Okay, there are help in different aspects of this work. Okay, so we're going to be talking about mosquitoes today, and everybody knows that mosquitoes transmit disease. Well, I was a little surprised to find out WHO came out with a report not too long ago and calculated, based upon human cases over the last decade, that fully 15 to 17% of all infectious diseases are transmitted by some sort of blood-sucking arthropod. Okay? So they're vector-borne. So I took it upon myself when I was preparing for this lecture. I thought, well, gee, I wonder how much of that is caused by mosquitoes. And some of the data I looked at is kind of sketchy on some of these more obscure vector-borne diseases, but by my estimate, it was a lot. 80, 90% of these diseases are transmitted by mosquitoes. Now, if we look just at mosquito-borne diseases, how many of these? There's lots of arboviruses that are transmitted by mosquitoes, but there's two diseases that predominate when you talk about mosquito-borne diseases in terms of number of annual cases, and that's malaria and dengue. So malaria and dengue are really different diseases, okay? Malaria is caused by a blood parasite, okay? And the clinical course of the disease can be very long-lasting and chronic, which is different than dengue. Dengue is a virus, okay? So the disease, the clinical disease is very acute, and you get very, very, very sick for a short period of time. Malaria usually occurs in rural settings. Dengue, usually associate that with urban settings. Okay? And they're two very different mosquitoes that transmit it. Okay, malaria is transmitted by this long, skinny mosquito that has long legs and spotted wings. It's the Anopheles mosquito. And dengue is transmitted by a dark-colored, kind of chunky mosquito that has brilliant white markings and banded legs. This is the Aedes mosquito. But both of these diseases have one similarity. Okay? Both malaria and dengue are what we consider anthroponotic transmission patterns. That is to say, the pathogen moves from human to mosquito to human to mosquito, and there's no involvement of other animals. There's no animal reservoirs involved with these diseases. So these are not zoonotic diseases. And that becomes important with what I'll be telling you next. Okay, so vector control, mosquito control has been a big part of the equation in trying to suppress or prevent transmission of these mosquito-borne diseases. And there's many ways in which we have this vector control, but if you look at them, you can see different strategies have evolved. Probably the most prevalent strategy, the one that has many more examples, is to introduce some insecticidal material out into the environment in order to kill the mosquitoes. And we're all familiar with aerial spraying of insecticides, there's larviciding. You can use perhaps a more targeted approach and incorporate that insecticide in fabrics and make bed nets and shirts and things. Less common, okay, or fewer examples, are to try to reduce the human-mosquito interaction or contact, okay? Such things as the use of personal and spatial repellents, okay? CO2 traps to trap the mosquitoes out. Even the use of air conditioning and television has reduced that contact, okay? Another strategy, a much more high-tech strategy, is to modify the mosquito populations so that they are, they're no longer reproductive, or they reduce their ability to transmit, okay? Such things as the release of sterile male insects, okay? Sterile male mosquitoes, or transgenically modified mosquitoes that are no longer competent to transmit. So there's a number of efforts underway. But what I want to spend the rest of my time on is a strategy that's not often used, okay? And that is, you could make the host toxic to the mosquito by giving it some sort of drug or insecticide. These are referred to as systemic insecticides. So someone takes the drug, and then when mosquitoes feed on them, their blood is toxic to the mosquito. And in terms of mosquito control for humans, there's really only one example, okay? We're down to one example, and it's the drug ivermectin, okay? Now, probably many of you have heard of this drug. This is actually considered by many to be a wonder drug, right up there with penicillin and aspirin, okay? And it's actually derived from soil bacteria, filamentous soil bacteria, okay? And the people that worked with it discovered that the extracts from this bacteria could cure mice of their intestinal parasites and their intestinal nematodes, okay? They actually won a Nobel Prize for discovering this. Well, the synthetic derivative of this natural product, ivermectin, okay, was marketed by the pharmaceutical company Merck back in the early 80s, and they marketed it as a livestock parasite treatment. And it soon became everywhere. Everybody wanted to have this wonder drug, okay? And then in the late 80s, Merck did a remarkable thing. They registered this drug, ivermectin, for use in humans to help combat two important filarial nematode diseases, okay, that were very intractable and hard to take care of. One was onchocerciasis, which causes river blindness, and the other one was lymphatic filariasis, which causes the horrible disfigurement of elephantiasis, okay? And so the World Health Organizations and other organizations and intergovernmental agencies to eradicate these diseases still today depend a lot on ivermectin. Well, a little bit more history. In the late 1990s, people were also looking at trying to combat malaria and eradicate malaria, but it started to falter. This effort was faltering due to, well, a number of things. Insecticide resistance was starting to happen, drug resistance by the parasite. And so people started to think, well, we need to have new tools to combat malaria. And there was a small group of people started looking back at ivermectin. And they found that ivermectin was very, very toxic to most species of Anopheles mosquitoes. And so as people began to talk about it and develop this idea, it soon was envisioned that maybe we could use ivermectin as a systemic insecticide on a community-wide basis as a complementary tool for malaria control. Okay? And so a consortium, a loose association of clinicians and epidemiologists and entomologists got together, and they developed a research agenda to what needed to be done to make this happen. Okay? They called it a roadmap. In fact, they call themselves the ivermectin roadmappers. And of course, the culmination of this effort by the roadmappers, of course, was to conduct large-scale, community-wide clinical trials to see if ivermectin could actually reduce incidence and prevalence of malaria. Okay? And these studies were very large in scale. They were all done in Africa. A lot of these studies, the results have just started to become published in the last year or two. And the very first one of these trials was conducted in the country of Guinea-Bissau. That's indicated by that red star. There was another large trial on the coast of Kenya in eastern Africa, indicated by the blue star. And then a really large trial was conducted in the island archipelagos off the coast of Guinea-Bissau, which is that yellow star. Well, as I said, these results have just recently come in. This is kind of what they found. I'll just real quickly point out that you can see that some of these studies were conducted for a single transmission season, some of them for 2 consecutive seasons. The oral dosing varied a little bit between these studies, but mostly they were talking about monthly dosing of people with ivermectin. Okay? And the size of the studies, some of them were quite large. The early studies were a couple hundred of people, several hundred people per treatment. Guinea-Bissau study had thousands of people taking these drugs and being followed for malaria. Okay. Well, and they looked at many things. They looked at the entomological, what was it doing to the mosquitoes? Were there any adverse effects on these people by taking ivermectin? But of course, the most important thing that they wanted to know was there a reduction in the incidence of malaria? In other words, the occurrence of new cases during the treatment period. And you can see that it was kind of a mixed bag, okay? Some studies said, yes, we've shown that there is a statistically lower incidence in ivermectin-treated communities. And others said, no, no difference, no difference. So the ivermectin roadmappers have really come to a crossroads. Some want to forge ahead, say, well, if we just increase the coverage, or maybe the different formulation of, and others are saying, no, forget it. Let's just go back to vaccines and bed nets. Forget it. It's not going to work. Okay? So one of the concepts that came out of, an important concept, I think, that came out of this agenda, this roadmap, was this idea of a mosquitocidal window, okay? Which merely means ask how long will treated people continue to kill mosquitoes after they take ivermectin. Okay? And of course, this depends a large part on the pharmacokinetics of the drug that they're taking. Now, the pharmacokinetics of ivermectin in humans had been known for a long time due to this effort to treat river blindness and elephantiasis. And this shows a typical pharmacokinetic curve of ivermectin at the standard dose that they're taking for the reduction or elimination of river blindness. Okay? So you see it goes up, and then it goes down. Now, the other element that's important when you're trying to determine or estimate a mosquitocidal window is what is the oral toxicity of this drug, okay, to the mosquito of interest. Okay? And that's typically done, the way that's typically done, an entomologist would use what's called a membrane feeder. Okay? A membrane feeder is just a little glass device. It's a cylinder. It's flared out at one end. And on the bottom, you cover it with some sort of membrane. You could be paraffin. We use sausage casing because it's go down to the butcher shop and get that stuff. And then you fill that cylinder up with blood. Now the cylinder, it has a little water jacket around it to circulate warm water to keep it warm. Okay, so that will attract the mosquitoes, and then you put it on top of the cage of the mosquitoes. The mosquitoes will detect the heat, they smell the blood in it, and if you're lucky, they'll go up and feed on it. Okay. Now this is a great system because this allows you to determine how much drug goes into those mosquitoes. Okay. You can set it up so you can deliver exactly the amount of concentration of drug that you want. And so you set up a series of concentrations in these membrane feeders, and you establish a dose-response curve. And from that dose-response curve, then you can just figure out what we call the oral LC50, the amount of drug that kills 50% of the mosquitoes. And I have a little picture here of Dr. Stacy Dreyer. Well, she's married now, Larson. She was in my lab, did a lot of these membrane feeds, and got really quite proficient at it. I relied on her for a lot for doing these membrane feeds. Okay, so, and with our mosquito, our Anopheles mosquito, we keep in culture over at Starcher Hall, is Anopheles stevensii. Okay, it's an important malaria vector in the Indian subcontinent, so that's one of your main vectors. Okay, well, Stacy had determined that the oral LC50 for that particular Anopheles mosquitoes was very, very low, 6 nanograms per mil. So it's very, very susceptible to ivermectin. So now if we take that oral LC50 and we were to chart it in that same pharmacokinetic curve, just put a little straight edge and just draw a line out where the 6 nanograms is, where those two lines cross would be the estimate of what your mosquitocidal window is. Okay, it's indicated by this shaded area. Okay, so when we did that, we saw, oh, mosquitocidal window is about 55 hours. That's like 2.5 days. What is that? That's not very good. That's not really good. So we weren't the only people that were doing that. The road mappers are doing that too. So one of the things the road mappers said, well, we need a wider window. Okay, 2.5 days is not enough. So we're going to have to dose people higher than we're dosing for this river blindness project. And so they ran some studies with some fairly large studies, and they said, well, what if we just double the dose to 300 micrograms per milliliter, and we gave it in 3 consecutive days? Okay. Well, when they did that, they could extend that window out to about 10 days. Okay. Well, I said, well, let's do it again. Let's double it. Okay. And we'll extend the window out to 15 days. But when they did that, at that dose of 600 micrograms per kilogram, they started to run into adverse effects. Okay, it wasn't good. So they had to pull back. So no, we can't go that high. Okay. So they're doing all this, and you know, by then I was starting to get involved with the ivermectin project. We had a little grant on it. We had a graduate student do almost her whole PhD thesis on it. And I was struggled with this idea, like, oh no, mosquitocidal window of only 10 to 12 days, is that even going to be enough? And I thought, well, maybe if we did this, or maybe if we did that. My head started to hurt, and I just thought, oh, I can't take this. I just want to go home. So I did. It was a beautiful summer day. I jumped in my car, and I did what a lot of people do. I just rolled down the window, and I cranked up the radio. And wouldn't you know it, the first song that came on the radio was one that I hadn't heard in years by the New Orleans icon, the late, great Dr. John. And I'll paraphrase, the lyrics go something like, I was on the right track, but it must have been the wrong train. Headed down the right road, but it was the wrong lane. And then as Providence would have it, the very next song that came up was the classic Huey Lewis song, I Need a New Drug. And it hit me, that's it. The road mappers, they're on the right track, but they need a new drug. What are these drugs? These are the azoxyzolene drugs, okay? And if you're a dog owner and you've taken your dog to the veterinary clinic recently, you may have encountered one or more of these drugs, okay? These are the latest generation in an oral flea and tick medication. So you give your dog one pill and no more fleas or ticks for a month, or even more, okay? So there's 4 of them out. The very first one to get FDA approval was a product called NexGard, and it contains as its active ingredient a compound known as afoxolator. Very soon after that, a second compound, Merck, jumped in the game, and they came out with a product called Brevecto. Brevecto, as its active ingredient, contains fluralaner. And pretty soon a third product showed up, Simparica, with the active ingredient here was sarolaner. And the newest of the batch has a funny little name, Credulio. Its active ingredient is laudanum. So what's the deal with these azoxystrobin drugs? Well, if we look at them and compare them with our old standby ivermectin, the first thing we notice structurally is they're a lot simpler. Okay? They're not as complicated, they don't have that big ring structure. And they're all built around this funny little structure here, this little ring structure that has an oxygen and a nitrogen, has these 3 fluorines coming off of it. Now, the variation in the products, of course, comes with the side chains. And I'm just showing the structure for fluralaner, but you'll see that fluralaner has, 2 chlorines and 3 additional fluorines. So these are, and the same with the other products, they're highly halogenated products. And they also work differently than ivermectin. Okay? The mode of action for ivermectin is that it binds tightly to the glutamate-gated chloride channels in the nervous system of arthropods and nematodes. And it has a broader spectrum of activity. It works against nematodes as well as arthropods. The azoxystrobin drugs only work for nematodes. They're not very effective against nematodes at all. Okay, so ivermectin, essentially, when it binds to these glutamate-gated chloride channels, okay, it locks those channels, essentially locks them in an open position so that chlorine leaks in, okay, and creates excessive inhibitory signals so that it overrides the excitatory nervous system. And what you see is the animal, if you feed this to the mosquitoes become very flaccid and fall to the floor of the cage, and they die. The death is very slow. It takes often days for these mosquitoes to die. Okay, quite different with the other drug. And the other drug, okay, its primary target is the GABA-gated chlorine channels. In this case, it closes them, it shuts them. Okay, and so it shuts off any inhibitory stimulation, the counterbalance that's needed to counterbalance excitatory nervous system. And so, without any break on the excitatory nervous transmission, these insects just become hyper excited and they drop to the ground, and within hours after feeding, they're dead. It's most remarkable. It's just, I've never seen anything like it. But the good part, the most important part, is the pharmacokinetics of these drugs. Okay, so when we do that, we probably should look at the dogs, okay? So this is some of the information that comes out of the dog heartworm literature, okay? Heartworm is another filarial nematode. It's treated a lot with ivermectin. That's a pretty high dose of ivermectin, okay? And you see the curve is a lot like what we saw with humans. There's a sharp peak and then a sharp, decline in the ivermectin in the plasma, okay? Compare that with fluralaner, okay, the different type of drug. First of all, you might notice that we're talking about a lot more drug. Okay, so the dogs can tolerate the higher concentrations of this drug. So you're going to get a lot more drug in the plasma. Okay, 105 times more drug. But what's interesting to me is that the tail off, the drop off in the drug is much more shallow. It's much more gradual. Okay, so you're thinking, this is very interesting. But remember, with mosquitocidal windows, it's not just the pharmacokinetics, okay? You gotta know something about the oral toxicity. Is it gonna kill mosquitoes? So this comes right out of Stacy's dissertation. She compared ivermectin with our mosquito, same mosquito, and fluralaner. And you can see the fluralaner is not as toxic as ivermectin, much less toxic. But how does that match up with the pharmacokinetics? Well, same thing. If you draw your little straight edge line out from where the oral LC50 is, we find that the mosquitocidal window in a dog, okay, against that species of mosquito theoretically would be about 7 days. But for fluralaner, single dose of fluralaner, you can stretch it out to, almost 2 months. That dog would be killing mosquitoes. Okay. So that looks pretty promising. Now, another way you could do this, Another way you could approach this is with a live host feeding. Okay? You could feed an animal drugs and then feed mosquitoes on them. And that's what we did. Okay? So we had a rodent system. We had these little paramyscus mice. And then we would take the commercial product, weigh how much is in there, look at the package to figure out how much drug is in there, and then we would dilute it according to the body weight of the mouse. Okay? And we mixed it in a little bait that was palatable to the mice. It was a little peanut butter bait. And you know, peanut butter gets all over the place. It's like, ugh, it's a mess. So we had to put it on little plates, serving plates, and edible serving plates of sliced carrots. They gobbled them up. They loved the drug. We didn't see any adverse effects in these animals. And then over time, we would do serial mosquito feedings. Okay, feed 20 or so mosquitoes on the mouse. Okay, put the mouse back in its cage for the next time. Go in the next day, come in and look at the mosquitoes and count all the dead mosquitoes. Okay. So we ran this with our Anopheles stevensii, and we had several different treatment groups. We had a control group. Each of these treatments, I believe, had like 4 or 5 mice in here. We had a control group that didn't get any drug. Ivermectin at a pretty high dose, and then 3 of the azoxyzolene drugs. Okay, so when we try to calculate that 50% mortality, we saw that, as predicted, the first one to drop out was ivermectin. Okay, and if you were to estimate a mosquitocidal window, about 5 days. So not much different than what we saw with the dogs. Okay. When we look at 2 of the drugs, fluorolaner and sarolaner, we were able, that mosquitocidal window was pushed out to 22 days. Okay. That's good. Lidocaine knocked it out of the park. Okay. This particular drug, we had, those mice were killing mosquitoes for 48 days. It was really quite amazing. So so it looks good, looks promising. We can get that mosquitocidal window out. But if you're a clinician, you're probably sitting there going, well, that looks good, but I have a question. Are these things safe for humans? Have they ever been tested in humans? Well, as it turns out, the answer is yes. There is one drug, happily it's the drug we're interested in, lidocaine, okay, has successfully undergone clinical trials in humans. Okay, when administered as oral capsules, so oral dose ivermectin. Okay, and this is just the summary of what you can pull off clinicaltrials.gov. Okay, these trials were done by a pharmaceutical company, Tarsus Pharmaceutical, and they have obviously looking to market these drugs for different purposes. And the main thing to notice is that they had a wide range of drug regimes. Okay, they did single doses, one-time doses from 25 to 600 milligrams. They did multiple doses where you take a drug and then you wait and take some more drug. And they did it for people that were on a full stomach and people that were starved. Okay, the bottom line is that the drugs were well tolerated. All of these dosing regimes. Okay. The only adverse effect that they saw was in a small percentage of people, there was headaches. Okay. And the headaches were not, importantly, the headaches were not in any way related to the dosing regime. So they were just like random headaches that people would get. Okay. So the FDA said, yes, go for it. You're FDA approved through phase 1. And now these drugs are being carried through phase 2 trials against ticks. Okay. Just in case you were wondering what these doses mean in terms of if you're a dog owner, okay, the highest dose of lidocaine tested, okay, that's Credulio, okay, is equivalent to the amount recommended for a mid-sized dog, a little border collie. That's the highest dose you're taking. Okay, so what I'm here for is to interface with clinicians and move this thing forward into some sort of phase 2 trial to look at the efficacy. Okay, so a very simple phase 2 trial might involve just a single dose, high dose of ivermectin and a placebo group. Okay, and then once you take the drug, you decide on when you're gonna sample. just take some blood draws, serial blood draws over time, and take that blood, and of course some of the blood you want to take over to the mass spec center and find out, how much drug is in those blood samples, so that you can figure out pharmacokinetics. But then the rest of the blood, you can take a portion of that blood and feed it to our Anopheles mosquitoes, targeting malaria. Okay. At the same time, you can take a subaliquot of the same blood and feed it to the dengue vector, Aedes aegypti. So we could say, well, is this going to work for Aedes? Is this going to work for dengue as well as malaria? Okay. One of the things that we have on this campus that a lot of medical places don't have,
Open the door.
why we can do this,
A prep room.
walk with me, come with me.
That's
Okay. In the inner sanctum, in the catacombs of Starcher Hall, we have this facility here. You see a lot of stainless steel sinks and little table, work table. We can do our membrane feeds. But the real guts of the operation is this area here, these racks. You see some screen cages and dozens of these little shallow pans containing water. And in that water contain maybe 200 mosquito larvae. Now, if you take care of them, and you feed them, and you watch them, they'll transform into the pupal stage. You got a couple of days, those people, you got to get them in a screen, because if you don't, you're going to have mosquitoes everywhere. So we have the capacity here on this campus to do that. If we can get clinicians interested to help me do these phase twos, we have the infrastructure that we can do it and move this thing forward. And again, the big picture here is what I'm hoping that we could move things forward is to use systemic insecticides in humans as a way to help control malaria and possibly dengue, okay, by adding these new drugs in with ivermectin. Expand the toolbox. And again, I think the crucial step at this point is to do some phase 2 human clinical trials to make sure this stuff works, okay? They've already been, at least that one drug has already been proved to be safe. Okay, so again, acknowledgements. These are the people that have helped make this presentation and helped make my research actually run, and I appreciate it. So I will stop there, and if there's any questions about mosquitoes or, dog drugs or anything, yeah. Okay. Yes, ma'am.
So one of the things that pops into my head, because there's a lot of interest in dealing with diseases and conditions that affect people locally. What about the vector for something like West Nile or?
Ah, yes. Okay, so as many of you know, you guys better know this, West Nile is not an anthropogenic disease, it's a zoonotic disease, okay? So you could envision, and West Nile virus is really a disease of birds, of songbirds, not seagulls or pigeons, but songbirds. So people have talked about, in fact, there's a guy that I know that's actually patenting the use of ivermectin in bird feeding. Okay, he says he's going to put out these bird feeders with ivermectin. We have not done that yet. We hope to do that this spring when the birds start coming in. We have the bird permits, we have the ICOOC protocols, we have the drugs, we have the mosquitoes, everything we need. We want to test, again, apply this concept of mosquitocidal window to songbirds. We'll probably pick a bird that we've worked with before, the grackle, because it's easy to maintain and it does well in captivity. And it comes to feeders, and it's a competent West Nile virus amplifying host. So yeah, so the concept could be expanded to that, as you know, and if everybody doesn't know this, I'll let you know that Dr. Brissett and I have been trying for years now to use these drugs for Lyme disease as abated incorporate them into rodent baits and put them out in the field for the rodent reservoir host of the spirochete, okay, to kill the ticks on the little rodents. We have one more chance at it. It could happen. I haven't given up hope yet that we can use these drugs for zoonotic diseases as well. That's a great one. Lyme disease is great. Tick-borne disease is perfect for these drugs.
Looking at your proposed and comparing it to that earlier study that was done using the ivermectin,
Yeah.
are you expecting a different result? So you mentioned the ivermectin. You sort of highlighted ivermectin in your clinical trial 2.
Yes. Ivermectin has gone through phase 2 trials for that sort of thing. And that's the whole idea behind a mosquitocidal window. So they were able to, at the dose that they chose to do those large scales, when they did laboratory mosquito feedings on the serum from those people that were received the drug, they were able to kill mosquitoes. Those people were able to kill mosquitoes out to about 10 days after receiving their last treatment. Okay, that's the mosquitocidal window. So what am I predicting from this? I'm predicting it's going to go out for a month, maybe even longer.
I thought the results from the African study were equivocal with ivermectin.
Yes, they were all, oops, sorry, wrong way. Yeah, I'll go back to that. These studies, what you're talking about, right? The big giant studies. That's all ivermectin, okay? The treatment dosings there, so they had treated in placebo groups. The treatment groups were all around 200 to 400 micrograms per kilogram body weight. Okay, that's what the dose was. And most of the studies, they would give them 3-day consecutive dose. So you get a dose of 300 or maybe 400 on day 1, day 2, day 3. And then you don't receive any more ivermectin until the next month. Okay, so they're doing that dosing regime once a month. And what I struggled with, I still struggle with, is if that dosing regime only produces a window of like maybe 10, 12 days, and you're only giving them once a month, that leaves another half of the month open that the mosquito will be able to transmit and take blood meals. So what we want to do is what ideally what you want to do is you want to make sure that you're killing mosquitoes for a month after one single oral dose. Wouldn't that be great? Dr. Brissett has enlightened, I got a little bit of time. Dr. Brissett has enlightened me a little bit. She's actually knows more about the Tarsus pharmaceutical people. So you've at least talked to some of those people, right? And Dr. Brissett told me that one of the delivery systems that they're looking at, and I thought it was kind of intriguing, was a transdermal patch, like a nicotine patch. So instead of nicotine or something like that, you just put in one of these drugs and then have it delivered. And I have no idea what that would do to the mosquitocidal window. I suspect that it would make it even better. But again, nothing's going to happen. Nothing's going to happen until we get those phase 2 trials done. Oh, now this is just, yeah. What I'm saying, this is probably the simplest thing you could do is just have.
Your phase 2 trial.
Yeah.
There was another slide to this.
I beg your pardon?
There was another slide that you proposed.
For isoxazoline or ivermectin?
Well, you had ivermectin on the next slide. Last slide. One of the last slides.
The last slide. There.
There. You got ivermectin here. Is that what you're going to be testing with ivermectin as well, or is this just?
Well, that depends on the scale of the phase 2 trial. The very simplest trial would be just to test that one drug at one dose. That's the bare bones. If we want to get complicated, sure, we could throw in ivermectin. It's already been done. I mean.
I guess I just misunderstood the slide. I thought ivermectin was one of the things.
This is the big strategy thing. Like, can we expand that fourth strategy of mosquito control by using systemic insecticides? I think ivermectin will always be there. Ivermectin is not going anywhere. It's a wonderful drug, but isoxazole, I can't even say it. The laners. So I had a friend of mine just say the laners. Just call them, lottolaner, foxolaner. The laners, right? I think they, in terms of mosquitoes, if all goes well, they will replace ivermectin. Ivermectin will always be used for nematodes, for heartworm, and all those other things. But these drugs, in my opinion, if we carry that far enough, they're going to replace ivermectin as a systemic insecticide for use in humans, okay? Because they're just better. Yeah. Thank you. I do too. I wish other people got as excited as I did, but yeah, I'm excited about it.
Yeah, I'm just wondering, did you look at the sex-specific, mosquitocidal effect, like male versus female also?
Say it again.
Did you look at the male versus female,
In the mosquito?
Yep.
Well, only the female will take blood. So we actually did goofily. I don't know if you guys even know this. Jumi and I were doing this. When these Anopheles feed, they actually drip a lot of blood. So we put in those dripping blood from these drugged mice, we put in male mosquitoes and let them walk around on the blood, thinking maybe that would be enough to transfer the material, and it didn't work. I think it could work. It might work, but
And other thing,
our little experiment, it didn't work.
so how many bites is sufficient to transfer the plasmodium? For example, like one bite is sufficient. So either way, before killing the mice, the parasite is already in the blood.
Yeah.
So
You need,
this is the
for
drug
both
also.
of those diseases, you need just one bite.
So even though my mosquito die, but they transfer the parasite. So does the drug also have effect on the parasite also, or
What is that?
the drug also have effect on the parasite also, or in the plasmodium, or?
There are people that are looking at that. I've looked at those papers. I'm not, people publish stuff and you go, yeah. And I'm looking at, I don't know. I wasn't convinced with people have asked the very same question. Does it work on the parasite? And there's one group that's saying, yes, it's working, but I wasn't convinced. I want to, this idea that maybe you can make a 3% difference, that's not enough. I want to see, boom, I want to see a big difference, Because these field trials that they did in Africa, they're fraught with all sorts of difficulties. The one I didn't show was from Mozambique. And right in the middle of the trial, a big giant cyclone came in and wiped everything out. You get, what can they say? All this effort. So it's not even worth talking about. So what I want to see is I want to see a big effect, like what we see. That's why we're picking 50% more. That's a big scale event, big outcome. I don't care about 2% or 5%. I want to see 50% or more. Anyway, I want to kill them. Anyway,
Jeff, I notice those compounds are all fluorinated. If I remember my biochemistry correctly, we have problems metabolizing fluorinated compounds in our bodies. I assume that the metabolism of these drugs has been characterized? Do you know?
not as well as you would think. If it has, I don't know about it. All of these drugs are commercialized, and there's a lot of proprietary things that we don't like. For instance, I would love to know what the pharmacokinetic curve was on those phase 1 trials with lottolaner, but that information is not publicly available. I mean, they have to, FDA will publish the general results of these trials, but they won't, You know, they're not obligated because the company, you know, there's other people trying to do this. They're in competition with other companies. So I don't
Part of the
know.
reason why the drugs stay so long in the blood is because of the fluorination.
Right.
It makes it hard. Do you have any idea whether drugs act as the active drug themselves, or whether they have to become activated in order to be?
Right. That's a great question. I do not know about isoxazoline drugs. I will say that with ivermectin, that's been shown to be the case. And we've seen it when we do, remember those crazy dairy things we did? Well, we repeated that with ivermectin. Forget about the mosquito vaccine, but we did treat them with ivermectin, and it kills mosquitoes. And what we saw, we've seen it, and there's a group in Thailand that's actually isolated some of the metabolites and shown that this is not with the drugs we're talking about, but with ivermectin. The ivermectin metabolites are actually as toxic or even more so than the parent drug, and that gets to the point is when we're doing these mosquitocidal windows and we're doing our little membrane feeds, we're actually working with technical grade materials. We get a little powder, a little vial of ivermectin, okay? When you do the live host feedings, of course, you're getting ivermectin, but you're also getting, as you suggest, these metabolites. And so we've seen that before in some of our early ivermectin studies, that the membrane, the IC50, in fact, we just published it, Stacy's stuff. The IC50, when we do a membrane feed with technical grade material, is like 6. But when you put it in an animal and you bleed that animal, we were doing this in cattle, and bleed those animals and take it over to the mass spec people, and they'll tell you, the IC50 drops from 6 down to like 2. Now, how is that possible? It can't be possible. The amount, and because what they're measuring is ivermectin. And so what we speculated was, yes, ivermectin, but those metabolites too are contributing to the overall, outcome of killing them. With these drugs, there's a lot to be done. We don't know. But for right now, I just want to get them. I want this thing moving. I want phase 2. So clinicians, help me. Help me. Anyway.
I have a couple questions.
Uh-oh.
One is like a practical endpoint thing. Like, if you did a trial in a village in Africa or someplace, like how many of the villagers actually have to be willing to take this drug, right? Because then as control, you're going to ask people to take this drug, on their own voluntarily, maybe for the rest of their lives, right? So how many people within a village or a population actually have to do this in order for it to kill the mosquitoes down to reduce the transmission of malaria?
Right. And to answer that question, I can't answer that myself. So what I do is I go to the modelers and I say, modeler, help me. What's the, and they will put together include pharmacokinetic models as well as transmission models, and hopefully have some idea that answers that question. And in fact, that's part of what Jumi's supposed to be doing. That's part of her PhD project, is start to model some of these things as soon as we get to a little bit more input data.
Because the idea here is that you're going to produce a protective blanket or a protective area within a population or a village or whatever, right?
Right.
Okay.
Right.
So my second question is, if this drug, let's say the mosquitocidal window is 58 days, how does that relate to the life cycle of the mosquito? Is this more than one cycle of mosquito? Or are you going to start to develop population effect within the mosquitoes that might actually be synergistic or additive with the drug effect?
Yeah. So the survival of these mosquitoes in the wild, probably no longer than, a 30-day-old mosquito in the wild. And these, I should say for everybody that not all mosquitoes are equal. Some mosquitoes are very short-lived. The ones we have around here, the vaccines, they don't live very long as adults. Okay, but some of these other ones that are important vectors they have a lot longer survival. I would say a 30-day-old Anopheles gambiae mosquito is a pretty old mosquito, okay? But they're the dangerous ones. The older the mosquito, the more dangerous, because the more time they've had to get infected and produce the parasites. So we want to get rid of these older mosquitoes if we can. I don't know if that answers your question.
I guess I'm thinking that you might limit the reproductive capacity of whatever the mosquito population is.
Yeah. Uh-huh.
If you kill the females, right?
Right. Right. Yes. Yes. And that's the one advantage that ivermectin has over these drugs, at least that's what we've done, okay? We look at these mosquitoes that survive their drug assault. Do they lay eggs? You know, can she produce a batch of eggs? And with ivermectin, there's a reproductive effect. The survivors of ivermectin, they don't digest their blood meal, their ovaries don't develop, there's something haywire with them. Okay, so we call them sublethal effects. Some people say there is, but in our hands, we've not seen this sublethal effects with these drugs. So there are limitations to these drugs. Yeah, but in terms of keeping people on drugs forever and ever and ever, I don't think this would be the design. I think this type of approach would have to be used with some of these other strategies. So it's going to have to be an integrated approach. We can't rely on that strictly. I was thinking about this the other day, actually, when I was making this talk. I think we can get this to work for dengue, and I'm sure it can. It'll kill those Aedes aegypti too. Maybe not as well, but it'll kill them. This kind of approach would be great for that disease because that disease, dengue, is very sporadic. And there's nothing happening. And all of a sudden, there's a dengue outbreak, okay? And in these cities like Rio de Janeiro and Jakarta, where this happens, they have a system. They have an infrastructure. They know it's right in that neighborhood. That's where they're coming from. At that point, that would be a great time to walk in with these drugs and say, hey, take these pills, take these pills, take these pills. Or if it's a patch, take these patch. And that would help quell it. Malaria, because it's rural, it's so chronic, I think it can actually be harder to do for malaria. I think it might be more useful for dengue. So maybe we better start rearing some 80s, huh? Start looking at that. Anyway, well, we have the capacity to do either one here on this campus, is what I'm saying. We're not afraid. We can do it. They're a lot easier than ticks. Ticks are so hard. Anyway.
Any other questions?
I want to thank everybody that showed up today, and I hope I stimulated some brain cells to think about different ways that we can kill mosquitoes. So.
On behalf of Transcend, thank you very much, Dr. Vaughan. That was great.
You're welcome.